of his research work he did in MIT. Last year when he visited Emily Brown group with Bob and other people, with Bob Haslinger and the others. So his work in progress, so it's very brief, I guess. And, but I think it's very interesting. Thanks, Miguel.
Hope so. Well, as Claudio said, this is actually a kind of report activity of my stay at the Massachusetts Institute of Technology, where I gave my first steps in neuroscience. Okay. I think we are blocking the first slide. Okay. So, understanding the mechanism for a neuron to make a spike is actually a very big problem right now. There are physical and chemical and biological mechanisms that are occurring inside the neuron. So it's till now it's not a very easy task. However, one of the tools that we have available in order to study how the neuron behave alone or in a population is to record the electrical activity of those cells. So, well, in this case, we have the spike train of one of the neurons. Till now, or till some years ago actually, single neuron recordings were only available. So, one of the things we can do with this information here is that since the spikes that we record are roughly the same shape, so we can discretize this information and binarize it in a time series. This time series can then be processed through mathematical object that is named point process, or in the way that we can use other statistical tools in order to study the spike train of a neuron. However, this happened only for single neuron experiments. After that, then we can have simultaneous recording of several neurons at the same time. For this case, in the beginning, this was seen as a way to make several single neurons experiment at the same time. But the actual power of the simultaneous recording is to record the activity of the neurons in a population at the same time. So not only to see one particular experiment over a neuron, but to see what's going on in all the population that we are recording. This raises the question that what should be the low-dimensional description that we can give to the spiking activity in a neural population? Well, one way that we can get this, we can give this an answer is to extract the dynamical coupling of the structure inside the population. In this case, what I'm calling dynamic coupling, or what I want to say effective connectivity, is different than the anatomical connectivity that occurs in the brain of an animal, because we are using only the observed spiking activity. So till now, or till the end of the experiments, we cannot get the actual anatomical connectivity that we are using. We have two frameworks in this work. The first framework is the experimental setup. The data was obtained with 4 rhesus monkeys. Craniotopy was made to the monkeys, and some electrodes were implanted in the way that we can access 3 different areas of the brain. Our electrodes were able to record the local field potential of the neurons around it. and the spike trains. In this case, the electrodes were, or in each branch, we have 96 electrodes, each one of them recording approximately one neuron at the time. The animals.
When you say recording approximately,
yeah, because
you don't know if you're recording a single neuron.
Exactly. You don't know. It's like in average, you have one or probably two neurons at the same time. The animals were asked, or yeah, let's say that were asked to perform two different tasks. One of the tasks was a learning task, and the second was a recalling task.
Sorry, Miguel. So coming back to the previous question.
Yeah.
So it means that you're recording some kind of local field potential. I mean, you're recording in the extracellular.
We're recording both.
Here, or you're recording. I mean, putting the microelectrode inside the neuron, because then in this case it's just one neuron.
Yeah. Well, there is something there. The electrodes are actually bigger than the neurons.
Okay.
So they can even cross the neuron, or they can be next to the neuron. This is something that we don't know in the moment that when the craniotomy is done. That's why we cannot ensure that we are recording one neuron in particular, or 1.5 or 2 of the neurons, because we don't know.
The point is that it could be more, because if the electrode is outside,
there is no more. The electrode is designed in such a way that maximum can get 2. Okay, so the animals perform two different tasks. One of them is a learning task, and the second is a recalling task. For the learning task, a blank screen with a small dot in the middle was shown to the animal at the beginning of the trial. This screen, in this screen appears a target. That it can be a figure, it can be a letter, it can be a number. In a particular position of the screen. There are only 4 different positions available: top, bottom, left, and right. After 500 milliseconds, which is the length of the epoch of each of this screen, we present a 500 milliseconds more delay, where nothing is shown to the animal. The second target is shown at the third epoch, in a different position than the previous one. And a second delay is shown. Then, after the second delay, we expect the monkey to react. The monkey, in order to give us his answer, has a joystick. And we will leave the animal with a blank screen, and with the joystick he has to perform to go from the center to the first target, and then back to the center and to the second target. So, if he does it correctly, then he will receive some rewards. There are some limitations in this kind of experiments, and it's the following. The monkey cannot react in the second delay time. If the monkey reacts in that time, then the trial is not successful. Also, we are not going to wait for the animal reaction for no longer than 2 seconds, which is the same time that is the length of the whole trial.
I presume one can design many experiments.
Sure.
Why this one? I mean, I would start with a single target and not this complicated thing.
Well, the thing is,
why this is important?
The thing is that the first target won't differentiate with the other task. So we need the second target to make the difference. In one of them, in this case, the animal is learning that the second one is here. This one is here, the second one will be in the top. In the second, that will be the recalling, this part will be different, the third epoch.
I guess you trained it without delay first.
Sure,
Right.
sure.
Okay. So you
The animals are trained to make this before.
And then you start with this new delay.
Exactly. Yeah. So, the second task, we start again with a blank screen. We present a first target, second delay, first delay, sorry. And instead of showing a second target, we're going to show a second delay. So there is nothing in the position of the second target. In this case, the animal has to remember where the second target was. So we are only talking here about short-term memory, because after a learning task, usually will come a recalling task. This is usually not always, because we don't want the animals to learn the pattern.
And in both tasks, the first target would be the same.
You can have two learning tasks in a row.
But if you have a learning task followed by a recalling task,
The first
the
target,
target has to be the same.
yes, exactly.
Always.
Yes, and the second target also.
Yeah.
The thing is that the animal won't see the second target. So, as I said before in the beginning, we can discretize the spike train of the neuron, and then we can construct a stereotypical event in time and study this through a point process.
How does the monkey know that the second is a recalling task and not a learning task?
He doesn't. And this is the thing. Once he sees the third epoch, that there is nothing there, then is the moment when he realizes that he has to recall what was the previous target. He doesn't have any information what kind of task he's doing until the third epoch. Okay, so we're going to use inferential statistic in order to try to get how the neurons are connected in the brain. Remember that in this case we are not talking about anatomical connections because we haven't finished the experiments. We are going to talk only about preferred or referred connections.
Excuse my ignorance. What's the point process?
Yeah, a point process is I think somehow you got me. It's a way to study some discretized phenomena. So when you have several trials of something, then you can get, for example, the statistics of that trial or along all these trials. This is what I understand of the point process. Okay, so the generalized linear models, we are going to use the logistic regression in order to get or to try to get the parameters that will fit the real data with the models that we want to do. We want to create models of the connections of the neurons. In order to do that, we are going to fit these kind of models for every one of the neurons in the population. So, in this case, the lambda represents the neuron's probability of spiking. And we are working in this case at millisecond resolution, which is the resolution of the spikes. Our first term represents the mean firing rate over the whole trial, so over the 2 seconds that last the experiment. Then the term F represents the firing rate, but now not over the whole trial, but over each epoch of 500 milliseconds. G represents the spike history of the own neuron. And H represents the effect of all other neurons in the population of the neuron we are going to fit.
Excuse me, maybe it's clear for you, but why you use this sort of representation? Why there is a logistic? Why you fit linear thing to the logistic firing rate?
Yeah, we are going to get the maximum likelihood.
But why the logistic? Why not another combination of the lambda rate?
Why not another combination?
I mean, why this?
This was the one we tried to get.
Yeah, but I can come up with 20 different functions that I can.
Yeah, I mean, the log likelihood will give you the parameters actually that fit better in the
But say
probability
this is
distribution.
better than another function.
No, well,
Say, let's take the Negroitan.
yeah.
This is something like a, well, no, this is something more of things.
Yes,
like the
exactly.
Negroitan.
It's just it's an statistical tool, and we just get it. We just use it.
But there is no.
Yeah.
I mean,
I'm afraid I cannot give a better reason.
I have a simple question.
Yeah.
And this is some equation in which you try to fit the data,
Exactly.
and where are the parameters?
Yeah, the parameters will be. I'm going to describe each one of these in a moment, and the parameters are
So
there.
you try to fit whatever data you have to that formula that comes from the wisdom of someone.
Exactly. So, well, in this case, what we did, we took the equation that is before that has 4 terms, and each one of the terms we call it a model. Each one of these models was a nested model. So we are going to fit the logistic regression from the simplest model to the more complex, to the most complex. Our null hypothesis is actually when we use the mean firing rate over the whole trial. This is the, let's say, the worst we can do. Is we are taking over the whole trial where at each 500 milliseconds something different is happening, we are taking nothing change. Over the whole trial is the same. So this will represent our null hypothesis. Our second model gives not only the mean firing rate over the 2 seconds, but also the firing rate over each epoch. This term here can be rewritten in this way, where the pi function are rectangular functions, like here, that has a length or is 500 milliseconds wide, and the height of each one of these rectangular functions is given by the fit parameter alpha that we are going to fit during the regression.
In the y-axis you have the frequency, the pulsating frequency.
Yes, exactly. We have the frequency of the spiking of the neuron. This will represent what we call, or what is called in neuroscience, a peristimulus histogram. The third model that we include has the history, the own history of the neuron. In this case, if the epoch for that particular neuron starts at 0, then we include what was going on before the epoch started. For the case that we model, we are going to include only information that is 50 milliseconds before the starting of the trial. In this case, this t history is the 50 milliseconds, and we have an indicator function that will indicate if between the time t and t prime there is a spike. If there is a spike, will be 1. If there is not a spike, will be 0. The second part of the equation is similar to the previous one. We have rectangular functions here and beta parameters that are going to be fit. For this case, the C functions are functions that last 25 milliseconds only.
And what is the shape of these functions?
They are square. They are rectangular like in the previous one. They are exactly in this way, except that instead of having a width of 500 milliseconds, they have 25 milliseconds. Because the history we are including is very short.
I have another question that I think maybe is related to our previous question of Maxi. So, in the right-hand side, you are just including the mean, the variance, the history, and so on, without any coefficients. So you are not, don't have a regression coefficient.
We are going
betas.
to get the betas and the alphas in the previous model, which will give us the shape of the probability distribution of the spiking. Well, the last model includes the population activity. So it's not only the neuron we are going to fit, but what's going on all around. In this case, the form of the last term is the same term than before, except that now we are adding over all neurons M, different than the particular neuron that we are fitting M. For this case, every beta is particular for each neuron in the population.
So I don't understand that. You are measuring one neuron.
I am feeding.
But how do you know the activity of the other neurons? You are recorded.
I have all of them.
And you have recorded all the neurons.
I have all the neurons in the population.
You have all of them.
Yes.
Six of them.
But I'm fitting one at a time.
But how many do you have? 6, 9? What was the number?
What, sir?
What was the number of neurons you recorded simultaneously?
Yeah, we can get between 50 to 96.
So you fit the nearby ones to the time.
If I am feeding the number 20, is all the others. I also have the spike trains of them.
Yeah, yes, how do you determine the spiking rate? I mean, the lambda is the spiking rate. How is this defined?
Yeah, this is through the
Through some resolution? Or?
Yes, yeah, you have to solve the equation for the linear regression.
No, you have the meaning of the data. How do you define the lambda of t? Lambda n of t for a single neuron. This is a probability.
Yeah.
And in a brain you don't have probabilities, you have voltages. How do you estimate the probabilities?
Because we transform that in a binary process. And then in the binary we just count the, it would be the mean firing rate. What do you mean?
No, I understand the mean firing. In the equations you have the left hand side and the right hand side. The right hand side you have the mean firing rate and the correction. But about the data which are in the left hand side, the lambda.
This will be given by the GLM, by the Generalized Linear Model.
No, the linear model what you have written. I'm talking about the data. How do you find the lambda of t? It is a probability, so you have to do average over something to get a probability. What is the ensemble of t?
The average is over all the trials that we're making the experiment.
Several trials.
Yes.
Okay, so the probability of the spike at time t is the proportion of times
of all the trials.
The neuron has a spike at time t.
Exactly.
Okay.
With a resolution, which is
1 millisecond.
1 millisecond in this case. So we use the log likelihood in order to get to differentiate the models or to select which models we are going to keep. We also use archaic information criterion that give us a comparison between different models. The problem with archaic information criterion is that it cannot reject your null hypothesis. So if all the models perform bad, he will only set which of those models is the less bad. But he won't say that this is the best in order to say it performs well. For the quality of the models, we use the Kolmogorov-Smirnov test that allow us to compare two different probability distributions. And we use also p-values. Since we are using more than two models, we are using the Bonferroni correction, that is actually the normal p-value, which is 5% or 0.05, divided into the number of models we are using. In a whole, what we did is the following. We have the data directly from the animals. We divided that data in 80% of the data to training and 20% of that data for testing. The 80% of the data was used for the nested model and the GLM. Then we construct all the models for each one of the neurons. We use now the model selection. We kept those models with higher log likelihood and that passed the archaic information criterion at least at 95% of accuracy. All the other models were discarded. And for the model that passed these two criteria here, we check the Kolmogorov-Smirnov test and the p-values. Only those that pass the p-value at less than 0.01, so at 1%, were kept, and only those that passed at 95% accuracy the Kolmogorov-Smirnov test were kept also.
By model, you understand a given set of values of the parameters alpha and beta that you determine.
Yeah.
That's a model.
Yes. For the different combinations that I put.
Yeah, if you put that sort of linear regression, you determine the coefficients alpha and beta from the data, and by a model, a good model is the one that fits the data according to all these successive tests.
A good model will be the one that, using the training data, I got some parameters, and then when I test with some data that is not seen in the system,
then it reproduces those parameters for
exactly.
those neurons using those values of alpha and beta determined for that.
Exactly. So the aim that we were looking here was actually to try to reconstruct the connections, the links between the neurons in the population. So in somehow the work actually finalizes here. We just get some of the different networks. In this case, I present how is the network when we present the target to the animal, the first delay, the second target, and the second delay. Till now, this doesn't have any meaning. It's just how the connections are linked, how the neurons are linked between them according to the inferential statistics we have done.
I'm sorry, I saw these links blue or red.
Yeah. So in the size of the node, in this case, represents the degree of the node, so the number of links that arrive, and the colors represent the clustering. This is the clustering coefficient for the particular nodes. But as I said, this is only a picture. Let's say we haven't studied this in using network theory yet. Something we did that was very slight.
But still, I mean, how do you determine the links? The links corresponds to parameters of your fit.
Could you go to the definition of the links?
Here.
No,
What are the links?
So just understand the meaning of the links. So
what are the links? Beta.
Beta is the link.
The parameters that are being fit.
So increase, it means that increasing rate is a positive beta?
Beta.
So you determine this beta QM, and if beta QM is different from 0, you draw one. Is that right?
Means that Q and M are connected.
There are only 2 betas for each.
What we see is actually we compare the probability distributions between the neurons. So if we see that one neuron has a probability distribution in some way, that we model it. And then with the testing data, we check that actually that probability distribution match, not only with them but with other neurons, then they are going to be linked. They are going to be connected.
You draw a link.
Yes.
So could you repeat again, please?
Yes. We compare the probability distributions between the different neurons in the population.
The distribution of what?
Sorry?
The distribution of?
The probability of the spiking.
The rate, lambda.
The rate, exactly. So, for those that share for example, the maximum log likelihood, then they are going to be linked in between.
So if neurons have the same probability distribution, you put a link between them.
Not the same, but there is a small threshold between them.
So if two
And the parameters are almost the same.
So if two neurons have the same value for mu, for the alphas and for the betas,
Not two neurons that spike simultaneously.
that would be that they have the same value for mu, for alpha, and for beta because they do the same thing.
Yeah, approximately.
So you determine. So
I understand that you are linking, you are putting the links, the two neurons are linked, not if beta equals, this was what I was thinking, is if the lambda resulting from the model at that particular time is the same. So they mean that they have the same probability of spiking at that time.
At that time.
So you link them if they have the model estimate that they have the same probability of spiking at that time.
So
Exactly.
if they spike simultaneously.
No, it's not simultaneously. You can allow some differences. You don't need exactly the same probability distribution.
With a given precision, if they spike.
Yeah, exactly.
But then question, why don't you construct this network from the real data without all this?
No,
Well, because
it's not linking when they spike at the same time.
No, but-
Even they have the same probability to spike at the same time.
Okay, so what
Exactly.
First you have to estimate the probability.
Right, but
This is what this is this.
But you don't need to construct this to estimate the probability. The probability is lambda, and you have the probability recorded for all the bands. So you can just compare if the probability of distribution lambda for a given neuron is similar to another one. You take this distribution, you take the other one, check the overlap, and that's it. And
Yeah, the thing is- the thing is that
Well, the point is why? But the point is, if what you want to reconstruct is this connection, why do you need a model?
Well,
A
because the
model
model can be useful later. But we have the model.
No, here you don't have a model. What is the model?
Well, there is no model. So it's a
Yeah, this one here.
Which a function, something.
Which a function, but why do you need that? If the only thing you want to determine if they spike at the same time.
This is
Well-
something I still don't understand. You record a series of spikes.
Yeah.
That lasts for 2 seconds. Each series of spikes for 2 seconds.
yeah, each trial.
And you repeat that how many times?
Sometimes 500 times, sometimes
You
200.
see, I thought you wanted to avoid that the monkey learn it. I mean, I cannot see.
Yeah,
how
these are very different
I
sessions.
cannot see how these measurements are independent of each other. I mean, the measurement number 100, there are 99 before, so the monkey has learned a lot.
Yeah,
The reaction is
it
not-
hasn't because the appearance of the targets are random. So the fact that the target appears in the left side of the screen doesn't mean that the second target will be in the top. So next time a left first target can mean a bottom in the second. So the animal is not learning that
the
pattern.
mechanics of the game. It's not
Yeah,
the-
he has-
It's not the same the first time you play this game or the 100th time you play it.
Yeah, but still you don't
When I
have.
play Tetris, when I used to play Tetris, I got better at it. Every time the pattern was different. But my knowledge of Tetris was better every time.
Yeah, but these are not how to say. These are not 500 trials in a row. This is not only a single experiment. This is done during a month or even longer. So where the monkey has some time to pass without playing with the joystick
The electron from
having
the monkey?
The electrons are forever.
for years actually.
They can live years.
Yeah.
So I have another question. You didn't say that, but it was written in your transparency that you were getting causality links. But you here we were talking about the sake of the probability, of finding probability.
Yeah.
It has nothing to do in principle.
What it means actually is what we say before is if you have one probability distribution that match with another one, this is what in this case we are calling causal.
But you can call it whatever, but it's really not measuring causality. I mean, why you say...
No, we are actually not measuring causality. We are actually not measuring causality.
Then why you call it?
Because one caused the other in the way that at least that the doctor said. I mean,
Who is causing who?
yeah, the direction we don't know. We just know that if one is very similar to the other, then they have the relation.
No.
Well, this is what we assume.
I can push these two guys and they have nothing in common.
They're sitting next to each other. Well, I don't know. Okay. So, just this is not our definite answer for the result. It's just to show something that that we are trying to get. What I'm showing here is the following. We have the comparison between the two tasks. So in green, in red we have the learning task, and in green we have the recalling task. What we see is that when we present the first target, the number of connections, this is actually the graph density, the number of connections that we infer doesn't change too much. They are very similar one to each other. When we show the first delay, there is not even too much thing. But once we show the third epoch, where we can have a second target or we cannot have a second target but a delay, then they differentiate a lot. Again, when we have in the last delay, then everything seems to be to go back to the normal. Still, this can probably be higher because the monkey is preparing to react with the joystick. We are not claiming right now that this will be like that, but we can infer from here is that the recalling task is a probably more difficult task that needs more connection in the population of neurons than a learning task. We still need more tests and more information to ensure that this result is true.
But the monkey doesn't know what he's doing, right?
No.
Then? I mean, why are you assuming at the epoch 3?
But we know what he's doing.
No, but the monkey knows. And the monkey is the guy who is doing the spreading.
But he has to react. He has to react whether there is. He has to react whether there is a target or not. So when there is not a target, he has to look in his brain. Where was the target in the previous task?
Well, but also for the other one, because he has to recover this information from moving the joystick.
In the other one, yes, but this is faster. This is 500 milliseconds long. In the other one, you can have even 3 or you can have 4 seconds between one task and the other. So in one, you have only 500 milliseconds to react. In the other, you have 4,000. So it's not exactly the same thing.
To clarify this, do you have separate sessions for what you call learning tasks and for recalling tasks? So you have just recalling tasks in a row, another day
no
tasks, or you mix them?
They have to be mixed.
So it's a presentation that is aleatory?
Yes, exactly.
So, following up on this, do you have any cue that indicates that it's a learning task versus a recalling task? I mean, you say you mix it, right? But is there any?
No, sure. In the moment that we are recording, we record what kind of task it is.
But it's some kind of stimuli that determine which is the phase, so to speak. Whether it's
no, it's just a random generator that creates the sequence of the task, and we know that generation.
And is that, I mean, it would change completely if you signal the session, so to speak, learning versus recalling. It would be a problem for your paradigm.
If you separate them.
So that the monkey knows
If you separate them completely.
If you provide a cue to the
stimuli, I mean, anything on the touch screen, whatever, that indicates that it's a recalling phase versus a learning phase.
Well, I think it could, because actually one of the things that we are thinking is that the animal in some of the tasks already know that after learning it most probably will come a recalling task.
Okay. So
and this is a pattern that
the sequence is always the same, like start learning, recalling, and then
no, you can have 2 learnings in a row. You can have 2 recallings in a row also.
So they have to learn to record what is going to happen. I mean, it's not always the same sequence.
Exactly, because we don't want that the animal learn the pattern.
Okay, I understand. Thank you.
Now, why do you call it learning? What is
Because, well, maybe it's not learning, but it's remembering. A very short thing.
It's a recalling task.
Yeah, it will be
short and long recalling.
Exactly, it will be something like short and long.
Yeah.
So here, then, even though that it might indicate that the monkey is waiting for something to appear on
Sure.
the screen.
In the first screen, yes, for sure.
That's why the recalling generates more activation, more neurons. Fire.
You mean because he expects to see something but he doesn't?
Exactly. He's got an expectation that is frustrating.
Yeah, it could be.
But that fits perhaps with should work with dopamine signaling, the operation of something interesting, or the lack of reinforcement when it should be there. I don't know if you have read about this. At least something similar or has nothing to do, I don't know.
So, if I understand, then what the link is that when you are expecting something, more neurons synchronize. Well, that's because it's the information is increased. That means that more neurons are doing the same thing
Because you get more
simultaneously.
attention raises, for instance. That could be.
So this is driving many.
It's driving the activation towards the goal.
Yeah, but it could be that different. I mean,
It could be something different.
it's just the right because you are wasting.
Okay.
They cannot do anything there.
For the test we performed, the GLM or the parameters we fit, they were in the 95% confidence level, indicating that the parameters we fit match with the data. They were not overridden. We have that the model 4, which is the most complex one, including the population activity, was selected 96% of the time, saying that usually those interactions that we are including were significant for the model. We need to perform more studies on these results. We have to measure some network properties to see what's going on inside, and we have, for the previous result that I showed, the preliminary result, we have to make some statistical tests of significance in order to see whether what we are seeing is true or is due to other kind of effects. Well, I was working with Professor Emery Brown from Massachusetts General Hospital, right here, anesthesiology, Rob Haslinger and Steve Williams from the MGHOs. Well, thank you very much.
No, Manuel me recuerda que los estudiantes de los I'm sorry. Manuel recalled me that the students are the first, so you are the next. Thanks. You are called with that. Is any student, PhD student, want to ask another question? No? Maxi doesn't qualify. Fernanda?
It's very interesting. Which region are you measuring?
Yeah, I don't know them by heart, but I wrote them. Here. We have prefrontal cortex, supplementary motor area, and dorsolateral prefrontal cortex.
Are three different?
There are three different electrodes, three different branches of these implanted in the brain at the same time.
Constantine?
You mentioned this archaic information criterion. Could you elaborate on this a bit?
Yeah, the archaic information criterion will actually is based on information entropy. So it will check whether when you have a set of models, it will check which of them is better, and it gives you that through something like the p-value, that you say, okay, if you pass or if you are lower than 5%, then this one is the one you have to keep. The problem that have Ike is that you cannot ensure that the model you are using is the best model. So it doesn't count for model quality. All the models you have can be very bad. Still, he will tell you, for all this model, use this one. But it won't tell you, this one is the one that rejects your null hypothesis, for example.
Right. Because, I mean, in your diagram, you had this pass and fail runoff, basically. So the fail would then basically be the 5%.
More than 5% error.
So this idea, let me say that the important point is that it weights the number of parameters. So obviously, if you have 100 parameters, you fit anything. And then this idea criteria tells you that if you have too many criteria, you penalize this one. Too many parameters, you penalize this one. This is the important point in the criteria.
So for me, the experiment is basically the learning task is you see something on the screen and you have to follow it with the hands. And then the recalling task is 2. You have twice directly following events with a short delay, and then you have 1 you have to reproduce with the hands with a time delay in the reverse time sequence. So in the end, it's learning the motor control and recalling both of them with a time delay in the opposite order. So did you change the delay between these two things as a parameter? I mean, for me,
the delay between one.
Okay, but why 5 seconds between the different experimental runs? Why not 30 seconds?
You mean the delay between a learning task and a recalling one? Yes, this is changing all the time. This is not fixed.
Okay, but it's fixed by the reaction time of the monkey, or do you control this?
This is controlled by the operator.
Okay, so it's not necessarily 5 seconds, it's sometimes also 30 seconds.
Yeah, not 30 seconds, not 30 seconds. But I think the
same order of magnitude.
Yeah, something like that. That you cannot wait too much between one of the other. Just remember this. I'm not a specialist in the experiment. I didn't do it by myself, but I think every second is money. Constantine?
No, Constantine.
I have a question really going to the heart of the motivation of this.
Well.
If you determine this network patterns that you get there, I can understand that it makes sense that you're able to distinguish or even see similar patterns and just make a classification based on similarity or dissimilarity of patterns. To that extent, I could follow. Beyond that, but is the motivation going beyond that, and I have the feeling it is, that you really want to interpret the particular network patterns, because that I would consider highly dangerous.
Well, we don't want the particular one, because the particular one will be the anatomical. We will have access to that one in the end of all the experiments.
But this gets a bit smoothed out and gets lost during the on the path, and in the end one has the impression this has to do something with a physiological network.
No, we don't have access to the physiological network. We don't know. We just know if, for example, loosely speaking, if this neuron spikes, and after that this one, then we'll make a connection. Probably that connection doesn't exist, or probably it does. We just, with the statistical tools, we try to give sense to that, to say, okay, more likely they have a connection. But we don't have access to the real one.
The language, they have connection. I don't know what that means. What does that mean?
The what?
What does that mean, they have connection?
Well, that they have,
functional relation is called.
Well, it's not even that, but in correlation, they're having the same distribution function is even weaker than a correlation.
Two Gaussians have the same distribution function even if they are independent.
Yeah, well, let's say that they have a similar behavior. This is what we are measuring.
I'm sorry, but I mean, my whole question is precisely going in that direction. So I'm missing the whole point, because if you just want to see whether the rates are the same in two different places, I mean, well, this could be a sophisticated way of doing that, but
it's not the spiking rate, it's the probability distribution of
and
the
what did you
spike.
get out of that? I mean, what have you learned? Because you are not interested in the physiology. I mean, you say that perhaps you can get it later. I don't think you can do it, but anyway. And so you are not interested in that.
There is a way.
So what is exactly the interest?
Now, we want to see whether the inferential links we are getting will actually be true. We can get the
Be true meaning that this being anatomical.
we can compare with the anatomics, but we have to wait till the end of the experiments, which is still haven't reached.
But in order to do that, basically it's a check of the method, so you can build a model.
Yeah, sure.
Play with a
Right now
and try to do that without.
Right now, through the
for doing these things. So, I mean, I suppose that there is something deeper there because you can do that with a model, right? To check that whether the method is really telling you something about the real.
well, this is what we have. We made the model, and then we now want to see whether that model actually gives the real thing, or give something that at least can reproduce what happens in with some errors, let's say.
He can also try to compare in the end of the experiment the functional connection to the structural connection and see if he finds some relation.
Yeah, it's not easier to build a model with a network and with some dynamics there and check whether the method is reproducing the anatomical connections than to play with a monkey and wait for some time to see whether.
Not always.
If this is the key, this is the goal. I mean, that's.
You have a question? Not anymore.
No, that's.
Okay, Maxi,
and probably this is not the easiest audience for this type of talk, but going one step beyond, I mean, all these establishing links. I mean, what are we trying to learn? What do we want to learn?
Well, in somehow, actually, what we just want to do, or at least what I understood, was to see whether the methods and the proposals that we are doing for the models are actually significant. To see whether those models can be used later. For that, then we have to apply and to reconstruct everything, and then to evaluate with the real connections.
But used for what? To learn what?
To see if we can have the model then for different monkeys or for different not tasks, but for different kind of experiment, then we can make some predictions at least, or we can have at least some previous. Some prior information before actually going to the experiment, which is.
But going against my deep way of thinking, I mean, why do we need these models for? We have the data.
Yeah, but we are not.
What do we gain from these models?
Generalization. This is my point of view. From the model, you can get some generalization because the data. Can be very different between one trial and another. So since you have this variability in the different, you are performing the same task, but there are different things what you are getting.
Reality, right?
So yes, exactly. But with the model, what you can get is generalization, because then if I give you just one trial, you are going to make conclusions that probably are not the correct one. But the model includes not only one trial, but includes a whole bunch of them.
But the model has no other content that it fits the data. So it is no better than the data.
No, it has a formula. He has not plotted, but I am plotting it. That's so. There's a parameter view, some rate that increase with the parameter view. I don't know if this has some meaning, but it's not. I work with biologists. They like very much statistics. Me and many other things don't like statistics at all. This is simply a model, and it's formulated in a way which is easier to use statistics to obtain the parameters. We physicists do it in a very different way. But essentially, you have a model, and then you try to fit the data. This is not so different as the statistics is something really strange to us. But that model probably is not so strange.
But there is that sentence that there are lies, there are deep lies, and then there are statistics.
I think it's a little unfair because to build the model with training data and then to try to predict what's going to happen in the test phase. No, that's what you told. So even if the model just fits the data into the data in the training phase, if the model is able to predict what kind of functional organization of activity, now it's at the functional level, then there's something
in another set of data.
The
Exactly.
problem is that it's limited by the areas you record.
Yeah.
Like you're recording this, you can only get maximum density of parameter related to this. But maybe the relevant connection is not between these areas, between the occipital or the orbital frontal, or whatever. So that's a limitation, but it's something, it's a way to develop the
like I think most of research, you have some constraints that you can keep your model inside it.
But what I wanted to ask you is whether you know about the Granger causality. Granger causality, I think it relates to the doubts about the skepticism based on the causal relations. There is a way to develop this sort of approach in a way that's not committed to a causal, physical causality, but just in informational terms, in which, well, when you have we take into account information relating to the activity of this area, of this network, of this node, you are eventually able to predict the behavior of this other node than just by the information related to that node.
Okay.
For what I know, is a very active paradigm in computational neuroscience in general.
It comes from economics.
In economics.
It comes from there.
Exactly. The economics doesn't come very much.
You have a question?
Yes, the take-home message. So, I understand that what you're trying to do is to identify functional systems, right?
Yeah.
I mean, not anatomically, but I mean, system or functional or whatever system in the brain that is working together to do any task. And it would be like a screening thing. So
not to any task, huh?
No, I mean, in this case, these are related also probably this area of the brain. But I mean, you can find out there are different systems inside the same area because they activate neurons, activate coordinated or synchronized in different parts, and they could be responsible for different tasks, whatever.
Yeah.
So we will like identify new connections functionally.
Yeah, exactly. But remember that we are working in a very tiny space.
Yeah.
So even the area that where is located the electrode can be much bigger than that. It can be something like this, and we are just in one point of that area.
Well, yeah.
So just to clarify, eventually the monkeys will be dead, and you will check the real connection, right?
Exactly.
And then you will be able to check whether the connections you infer are actually anatomical.
Yes, through some other neurons. The electrodes are separated in such a way that they are not registered in the same neuron at the same time. So they are not even, they are 400 microns. So there could be some neurons in between.
But the goal will be that, in the end, will be to check whether the links you infer, the functional links you can deduce, actually correspond to anatomical links.
Yeah, or at least mostly correspond to the anatomical ones.
Last question. Or not?
Up to which extent do you think the electrodes, or does the presence of the electrodes disturb the activity of the neurons?
Definitely it does.
It does? Okay.
Yeah, because it can happen that the electrode cross one of the neurons and it destroyed it in the moment that it does. However, from the moment of the...
Maybe that they simply avoid using the brain region or something?
Yeah, from the moment that the craniotopy is done, so the electrode is implanted, you have to wait some time for this kind of neuron even to die or to recover.
But it's not clear to me what is the role of the part of the heuristic. I mean, all the results you show is for the part that you show the signals, and the difference is in the part that you show the signals. So, what's the part of the monkey playing with the heuristic? I mean, why that part? You can skip altogether that, or?
Yeah, the data have to match with those tasks that were actually well performed. So it doesn't work for us if the monkey doesn't react. For example, so I need the answer of the monkey.
So you only keep the data or depends in which the monkey react properly.
Correctly.
The other ones are discarded.
Exactly.
Okay. And now the other question: why 4 monkeys? And what's the difference between the different monkeys? I mean, do you take the data all together, or you separate by individuals, or?
They are analyzed individually, even across experiments. So the same monkey doing the same task, but in different sessions.
Yeah.
And after that, they're average per monkey.
But when you plot this data where they are showing that, for example, the variability...
This is actually the average of the average.
The average of...
Across session, across monkeys.
Constantine, last one.
What's the variability between sessions and meeting monkeys? Do you know?
What do you mean with the variability? The variability of what?
The variability of the model, I mean the fits of the model that you are presenting, or the accuracy of the model to predict what you see in the experiment.
If you keep the animal, the same animal, the variability is not so high. I cannot tell you the number right now, but the variability is not so high. Now, when you go through animals, this increases. That's why you have these error bars in the end that are a little bit wide.
Well, okay. We have the coffee break. Thank you again.